Hi everyone, as promised, this is your mini lecture for Wednesday this week. To round out the first sort of third of our class, I want to address something that every biologist spends a huge amount of time doing, and that is designing experiments and communicating the results of your science. We've already looked at a lot of figures this term, and we're going to see more and more of them. Moreover, you are going to be expected to be able to make really nice figures in papers and in lab write-ups that you're doing for this class and for others. So we're going to spend some time first reviewing experimental design considerations, some basic statistics, and then learning to make really nice, clean figures and to interpret the data that are displayed in the figures we make. On our first day of class, I told you a story about acorns, mice, ticks, and Lyme disease. I'm going to use this as an example for the next couple of slides, so let's review what was going on. Remember that if we have an acorn mast in one year, like 2010, we get more mice in 2011. Young ticks do really well because there are plenty of mice for them. Then the next year, there aren't as many acorns for the mice to eat, so mouse density drops. But there are lots of adult ticks around. They've now grown up, and they have something to eat. But that is humans. Humans become a more likely target, so Lyme disease increases in abundance. In ecology, we ask lots of different kinds of questions, and our answers to those questions take on lots of different forms. Natural history observations, the analysis of patterns, experiments, and quantitative models. On the first day of class, we talked about Richard Ostfeld, and we said that he made a really critical natural history observation in this system, that white-footed mice get ticks on their ears too. Recall that we first considered deer populations most important, but realized that deer were a really poor reservoir, so that couldn't be what was going on. Ostfeld also employed the analysis of patterns. For instance, he statistically assessed a pattern between acorns and mice and found that when red oak acorns were abundant in one year, that mouse density increased the following year. But better yet, what if Ostfeld actually did controlled experiments that would help us to assess whether something was the cause of an observed pattern? We could imagine that Ostfeld might do something like manipulate the amount of acorns in different areas and ask whether the density of mice increased in the year after in areas where acorns were more abundant in year one. So let's pretend that these boxes are showing you three plots that he might have used in this experiment. In one, he didn't add any acorns. In one of them, he added twice the number of normal acorns, and in another, he added 5 times the number of acorns that you would encounter naturally. We want to think here about which of these is a manipulation and which is a control treatment. So if you recall from earlier classes, the way to think about a control versus a treatment is whether something has been manipulated or not. So our no manipulation here is the situation where there are no acorns added, and that'll be the control that we're making comparisons to in this case. But in this experiment, we have to worry about things like outliers and confounding variables. What would happen, for instance, if our 5 times natural acorns area was also an area where there was a disease that wiped out a bunch of mice, but that we didn't know about? In this case, our pattern might be really confusing because 2 times the natural acorn level would lead to more mice, but 5 times the natural acorn level would lead to fewer. In other words, we'd end up with a hump-shaped distribution, even if our true relationship was a positive and linear one. So what is it that we could do to improve on our experimental design that would eliminate that problem? Well, he could have replicated this experiment, right? So that each treatment type occurred multiple different times. And that's an awesome idea, but there's still a problem with this experiment. An even better experiment would include randomization. Ostfeld could randomize the treatments across localities. And why would we want to do that? Well, we'd want to do that because it reduces bias by equalizing other factors that have not been explicitly accounted for in the experimental design. So again, imagine that now we have this disease at locality 3 over here on the right. In this case, the disease ends up being spread across all 3 treatments rather than just the 5 times acorn treatment. Ecologists also use quantitative models to make predictions about what might happen in a particular case. You might be familiar with these types of things from weather and climate modeling. For instance, what would happen when the winds were just like this? We've seen a lot of these sort of predictions recently, unfortunately. And these models are based on a huge amount of previous knowledge and make predictions about the future. We haven't gone into much detail here yet, but you could imagine that Ostfeld might be able to develop a model that would enable him to predict future acorn masting years, and from that to extrapolate to Lyme disease incidence based on all of the long-term data that he's collected on the ecological relationships involved. So what do I want you to take home with you today? I want you to take home the idea that ecology is a science. We make observations, we ask questions, develop hypotheses, and evaluate them through observations, experiments, and the use of quantitative models. And then we modify our hypotheses when they aren't upheld in our analyses. We pose new questions and we start over again. It's a process that's iterative and self-correcting. So these methods that we've just talked about are the very things that ecologists think about on a day-to-day basis. How do we design experiments that allow us to answer our questions and address our hypotheses? Next, what I want to do is spend a little bit of time talking about that part that involves evaluating your hypotheses, measurements, and statistics. So what are the tools that we typically use? Well, we usually have some kind of a question in mind when we go out and take measurements. Something like, what's the average value? Is there a trend? Is there a difference between these groups? What's likely to happen in the future? And ecologists love to count and measure things. So here are pictures of folks who are out in the environment measuring different ecological variables. But it's often much too difficult to measure an entire population. So instead of measuring every single individual in a population, we sample populations and extrapolate from those samples to the population on the whole. So imagine that we wanted to know something about all Americans. How much do they weigh? How tall are they? What proportion considers themselves to be Republican? How many miles did you walk today? It takes way too much time, energy, and money to ask every single American these questions. The answers to all of these questions have some kind of distribution, and their average is assumed to have some true value. We call that true value mu. So how do we figure out mu? We don't. Instead, we sample a subset of the population and we estimate something that we call X bar, which is just the mean for the sample. So usually when we're sampling a population, we're concerned about 3 different types of metrics. We want to know something about the central tendency, something about the spread of the data, and something about our confidence. In other words, how certain we are about our estimates. So on the next couple of slides, I want to talk about how we measure 2 of those, the central tendency and the spread of the data. So let's start with measures of central tendency. There are 3 measures that we typically talk about: the mean, the median, and the mode. And we're all most familiar with the mean, probably, the average, right? But there are reasons to use other measures of central tendency, like the median and the mode. And you'll have to really think back to elementary school to remember those. So first off, recall the way that you calculate the mean. You guys are going to practice that with J. And I want to remind you about these other 2 things. Anyone remember the mode? The mode is the most common number in a series, and the median is usually referred to as the middle number of a series. But remember that if you have an even number of observations, you have to take the mean of those 2 middle numbers in the series to get the median. But why don't we always just use the mean? Why bother with those other things? Well, we can illustrate this point by pointing out that if we had one plant on this distribution that had 5,000 leaves, it would push our mean all the way up to 718, right? So if our original mean on that last slide is 5, our median is 4.5, and our mode is 4, if we add just that single value way up at 5,000, that pushes our mean all the way up to 718. But notice that the mode would actually stay the same. There are still 2 plants with 4 leaves, and the median would only get bumped up to 5 rather than 4.5. So the key here is that the mean is really sensitive to extreme values, whereas the median and the mode tend to fall in the center of the distribution regardless of its shape and spread. So how about measures of spread? The two that we'll talk about and practice calculating are variance and the standard deviation. And you can see here that these two things are actually related to one another. The standard deviation is a measure of how much spread there is from the mean. In this figure, each line represents one standard deviation from the mean. So if you compare the normal distribution on the left-hand side of the slide to the one on the right, you're looking at distributions that differ in their spread or variance. The figure on the left-hand side has a larger standard deviation. So if you have a large standard deviation, it might look something like this. And with a small standard deviation from the mean, the distribution might look like this sort of scrunched up one on the right instead. So on the right, small standard deviation, our data points tend to be close to the mean, and on the left is a large standard deviation. Our data points tend to be spread over a large number of values. So you're going to practice calculating variance and standard deviation with J today, and you're going to do that using this fun data set that I'm going to tell you about now and that J will reiterate. We're going to put this all into practice with a really simple example. So perhaps some of you have heard of something called a pitfall trap. A pitfall trap is basically a cup that you place in the ground at ground level, and it either has alcohol or soapy water in the bottom of it. And as arthropods, like the one that is walking across here, walks by that pitfall trap, they fall in. So these are commonly used to estimate arthropod abundance and biodiversity in the field in ecological experiments. It's super simple to do, and there are lots of different types of pitfall traps that you can use. You can have really fancy ones that trick insects. You can buy them pre-made. These are just a few examples. The one on the top left only captures ground-dwelling arthropods, not flying ones. The long one off to the right is a trap that's used to trap insects that are coming from a specific direction. And then on the bottom left is just a yellow solo bowl. This is super cheap, and it collects both ground dwellers and flying arthropods. And these are by far the most popular and the most cost effective. But here's the problem. They're actually endangered. Solo Plastics now only supplies red and blue bowls to grocery stores. You actually have to go to Party City and buy the really expensive yellow bowls, and no one has time for that. And we know that ecologists are notoriously cheap about our experiments, so we don't want to make them spend more money. So a group of teachers who were visiting DU for some professional development have actually been conducting a study looking at whether yellow bowls really are any different from red bowls or blue bowls in their ability to attract insects and to collect them. So this is our question. We want to know whether the trap color influences our estimates of insect abundance. And you'll be working with the data set that I'm providing here on this slide for most of the class period when you're in class. So these are the values that you'll use with J to practice calculating means, standard deviations, variance, and then to practice making really lovely graphs. So here's your summary slide for the day. Have a great time with J, and best of luck to all of you on your exam tomorrow. Remember that for this particular lecture, you'll only be tested on very general things today on this exam tomorrow. So concentrate your efforts on things that we've talked about prior to today, but make sure you've actually watched this ahead of time. Thanks, everybody. Talk to you later.